let's start with a really fundamental problem that every single cell has to solve. Okay, picture this. You've got a bustling city. It needs walls, right? For protection, to keep everything organized and safe inside. But a city that's completely sealed off from the world, it's gonna die. It needs roads and gates for trade, to bring in supplies, export goods, and get rid of all the trash. Well, a cell is no different. So how on earth does it solve this paradox? How does it build a wall that is also a gate?
Well, the answer, of course, is the cell membrane. And let me tell you, it's so much more than just a simple bag. It's this dynamic, intelligent, flexible barrier that is constantly sensing its environment, communicating with other cells, and meticulously managing all the traffic of molecules. This structure really is the hero of our story today, the silent workhorse that makes all cellular life possible.
To really get our heads around this, here's our roadmap for today. We're going to start by building the membrane from its core components, something called the fluid mosaic model. Then we'll explore the different ways things get across it, both the easy ways and the hard ways, passively and actively. We'll look at some real-world examples and cool adaptations, and then we'll finish up with some key takeaways and exam tips to make sure you've absolutely mastered these concepts. Right, first up, how is this incredible structure actually built? We can't really understand what it does until we appreciate what it looks like. So let's break down the architecture of the cell membrane, which scientists have this beautiful description for, the fluid mosaic model. Okay, the foundation, the main fabric of it all, is something called the phospholipid bilayer. Now, each of these little molecules is kind of two-faced. It has a phosphate head that is hydrophilic, meaning it loves water, and two fatty acid tails that are hydrophobic, meaning they fear water. So what happens when you put millions of these in water? They just assemble themselves. The heads all point towards the watery environment inside and outside the cell, while the tails desperately hide from the water, creating this non-polar oily core. And that core is the fundamental barrier that stops water-soluble things from just drifting in or out. But a barrier isn't enough. We need the gates. And that's where proteins come in. They're studded all throughout the membrane, and they are the functional workhorses. You've got integral proteins that span the entire membrane, acting like channels or carriers. Others are receptors that bind to hormones. Some are even enzymes. Then you have peripheral proteins that just sit on one side. And the importance of these proteins, it just can't be overstated. For example, cystic fibrosis is caused by a mutation in the gene for a single membrane protein that's supposed to transport chloride ions. When that one tiny gate fails, it has absolutely devastating system-wide consequences. Now, in animal cells, cholesterol plays this really fascinating role. It wedges itself between the phospholipid tails and acts like a fluidity buffer. You can think of it as a chaperone. If the temperature gets too high, cholesterol's rigid structure holds the phospholipids back, stopping the membrane from becoming too fluid, kind of like melted butter. But if it gets too cold, cholesterol gets in the way of the tails packing too tightly, preventing the membrane from becoming a rigid, fragile solid. It is absolutely crucial for keeping that perfect level of integrity across a whole range of temperatures. Then, on the very outside of the cell, you have these short carbohydrate chains attached to proteins and lipids, and together they form what's called the glycocalyx. The best way to think of this is as the cell's unique molecular ID badge, or its fingerprint. This is how your immune system recognizes your own cells, friend, from foreign invaders like bacteria, foe. And a perfect real-world example of this is your blood type. The A, B, and O antigens that determine your blood group are just different carbohydrate chains on the surface of your red blood cells. So why do we call it a fluid mosaic? Well, mosaic is pretty simple. It's because it's a patchwork of all these different components we just talked about. And fluid is because these components are not cemented in place. The phospholipids and most of the proteins can actually drift around laterally, kind of like people mingling at a party. And this fluidity is essential. It lets the cell change shape, it lets it repair itself if it gets punctured, and it lets proteins move to wherever they're needed to do their jobs. It just brilliantly shows that the membrane isn't a static wall, but a bustling, active environment. And the degree of this fluidity is also affected by the phospholipids themselves. You see, if their fatty acid tails are saturated, they're straight chains that can pack together really tightly, making the membrane less fluid, more viscous. But if they have unsaturated tails, those tails have double bonds that create kinks, and these kinks push the phospholipids apart, which makes the membrane more fluid. Organisms that live in really cold places, like Arctic fish, will actually have more unsaturated fats in their membranes to keep them fluid and functional even when it's freezing cold. Okay, I know that was a lot of components to remember, so here's a really handy way to lock it all in your brain. Just remember P, G, C, F. P for the foundational phospholipids that create that bilayer, G for the glycocalyx ID tags on the outside, C for the cholesterol buffer that maintains fluidity in animal cells, and F for the overall fluid nature of this whole mosaic model. PGCF. It's a great little trick to recall those 4 key structural features. Okay, so we've built the membrane. Now let's see how things actually get across it. We'll start with the easy way, passive transport. This basically means moving substances without the cell having to spend any of its precious energy. It's all about simply going with the flow, down a concentration gradient. So there are 2 main types of passive transport. The first is simple diffusion. This is for small, nonpolar molecules like oxygen and carbon dioxide. They're like tiny spies that can just slip right through that oily phospholipid core, moving from high concentration to low. But what about the bigger or charged things like glucose or ions? They get repelled by that oily core. They need some help. And that's where facilitated diffusion comes in. It still doesn't need energy. It still moves down the concentration gradient, but it uses a specific protein to help it along. Some are channel proteins, which are like simple tunnels, and others are carrier proteins, which actually change shape to ferry the molecule across. And then we have this special case of osmosis. Really, this is just the net diffusion of water across a partially permeable membrane. Now, with water, we use a special term, water potential. Water will always move from an area of high water potential, which means it's less concentrated with stuff like salts or sugars, to an area of lower water potential, where there are more solutes dissolved. And this has huge consequences. An animal cell in pure water will swell up and burst, but a plant cell, thanks to its strong cell wall, just gets firm and turgid.
All right, but what happens when a cell needs to pile something up inside or pump something out, moving it against the natural flow? This is like trying to push a boulder uphill. It takes focused effort, it takes a ton of energy, and it's absolutely essential for things like nerve impulses and absorbing all the nutrients from your food. This is active transport.
So the crucial point here is this. Active transport moves things from a low concentration to a high concentration. And to pull this off, it needs 2 things. First, a specific carrier protein, often called a pump, which is super selective for its target molecule. And second, it needs energy. This energy comes from breaking down the cell's universal energy currency, a molecule called ATP. Think of ATP as a tiny charged-up battery. When the cell spends it, that ATP is broken down, releasing a packet of energy that powers the pump. No ATP, no active transport. It's as simple as that. Now, cells can get really, really clever with this. Take co-transport, for example, which happens in your small intestine when you're absorbing glucose. First, a pump uses ATP to actively push sodium ions out of the gut cells. This creates a really low concentration of sodium inside the cell, which sets up a steep gradient. Then, a different protein, a co-transporter, takes advantage of this. It lets sodium flow back in down its gradient, which is an energetically downhill process. And the energy released by that is used to pull a glucose molecule along for the ride, even if it's dragging that glucose uphill against its concentration gradient. It is a brilliant biological 2-for-1 deal. So to keep all these different transport methods straight, here's our second mnemonic for the day. DFOAC. We've got D for simple diffusion through the bilayer, F for facilitated diffusion using proteins, O for osmosis, the special case for water, A for active transport, which needs that ATP, and C for that clever co-transport that uses one gradient to power another, DFOAC. That covers all the bases for getting things across the membrane. Okay, so we've covered the theory. Now let's see how this stuff actually plays out in the real world. How do cells specialize to become absolute masters of transport, and what external factors can change how fast all of this happens? Well, cells that are specialized for absorption, like in your gut or your kidneys, have a few key tricks up their sleeve. They massively increase their surface area with millions of tiny folds called microvilli. Get this, the surface area of the human small intestine is about the size of a tennis court. They also just pack their membranes with a huge density of carrier and channel proteins. They work tirelessly to maintain steep concentration gradients, for instance, by having a rich blood supply to immediately whisk absorbed stuff away. And finally, the exchange surface itself is incredibly thin, often just 1 cell thick, to make that diffusion path as short as possible. This table sums up the factors controlling the rate of transport really nicely. A steeper gradient, a larger surface area, and a shorter diffusion path all increase the rate. Higher temperature also boosts the rate because all the molecules have more kinetic energy and they're moving around faster. But, and this is a big but, this only works up to a certain point. If it gets too hot, those crucial membrane proteins will denature. Their specific 3D shape unravels and they stop working, causing the transport rate to just plummet. And of course, for facilitated or active transport, the rate is ultimately limited by the number of protein helpers you have available.
And, this isn't just theory. It's something we can actively measure and investigate right in the lab. A classic practical experiment involves figuring out the water potential of plant tissue, and it's a fantastic way to see osmosis happen right before your eyes.
Here's how you do it. You create a series of solutions, maybe sugar solutions with different known concentrations. You cut identical pieces of a plant, like a potato, and you weigh them very precisely. Then you put one piece in each solution for a fixed amount of time. Afterwards, you take them out, carefully blot them dry to get rid of any excess water on the surface, and weigh them again. By calculating the percentage change in mass and plotting that on a graph, you'll see a trend. In the solutions that have a higher water potential than the potato, the pieces will gain mass. In the ones with lower water potential, they'll lose mass. And the point where your graph crosses that zero change line, that's the isotonic point. That's where the water potential of the solution is equal to the water potential inside the plant cells.
Okay, let's bring all this home. To truly master this topic, especially if you have an exam coming up, it really helps to know the common pitfalls and the key phrases that show you really deeply understand the material.
Here are some classic mistakes you should try to avoid. Don't just say diffusion if you can specify whether it's simple or facilitated. Be precise. Always, and I mean always, mention ATP when you're discussing active transport. It's totally non-negotiable. For osmosis, you've got to use the correct term, water potential. And if you see a graph of transport rate that flattens out, don't just say it stops. You have to explain why. It's probably because all the available carrier proteins are busy. They're working at their maximum capacity. They become saturated. And finally, always try to link a cell's features, like those microvilli, directly back to how they help its function.
Want a sentence that just screams, I know what I'm talking about? When you're describing active transport, try this one. ATP is hydrolyzed to ADP plus an inorganic phosphate. The energy released is used to change the tertiary structure of the carrier protein, moving the substance against its concentration gradient. Every part of that sentence shows a deeper level of understanding. Hydrolyzed is the specific reaction. ADP plus pi are the products. And changing the shape is the precise mechanism. That right there is a top-tier answer. So we'll end with this thought. We've seen how a structure that's just a few molecules thick is actually this dynamic, intelligent, self-assembling system of gates and sensors and pumps. It actively and constantly works to create a little pocket of order and a universe that always tends towards chaos. So if a single membrane built from components that are themselves lifeless can show this level of complexity, it really makes you appreciate the elegant, emergent properties that define life at every single scale.